Coming to the next one, why Pisum sativum? Why Mendel worked only on Pisum sativum and no other plant? First point was that it was easy to cultivate. In his monastery, and imagine in those days, he didn't receive any funds from the government. There were actually no funding at all for his project. So he was working on something at his backyard. And when he was, doing this process, he didn't even realize what kind of work he is going to do for the world. So the first thing was, it was easy to cultivate. And the life cycle of pea plants is very short. Within a year, he could actually grow some 3 generations. And the third point is that he could see distinct heritable differences. Like I told you, either tall or dwarf, either black or white. Okay, so for you to understand, I'm saying. So he was able to see that either this or that, there is only extremes and there is no intermediate characters at all. And number 4, it was easy for self and cross pollination. When he was doing all this hybridization experiment, he was actually sure and he was finding it very easy for doing the process of self and cross pollination. Now let us see what were the 7 characters chosen by Mendel in pea plant. The first character was plant height. Second was flower color. Third is flower position. Then pod color. Then pod shape. And then seed shape and seed color. These are the clear-cut different extremes of characteristic features he was able to observe in Pisum sativum. What is the first one? Plant height. Either the plants were tall or they were dwarf. Flower color. They were either purple or white. That's all. There is no intermediate color between these two. And flower position, axial or terminal. What do you mean by axial? The plant would end up in a, this is terminal. Plant stem, the main stem would end up in a flower. That is terminal. When I talk about axial, the flowers are borne on the lateral branches. So this is axial bearing of flowers is called as what? It is a dominant trait. Another one is terminal, which ends in a flower. Now, I will teach you later what is dominant and recessive trait. For now, you understand that these are the characteristic features. Pod color. So, pod color means the outer covering of a pea pod. This can be either green or yellow. Pod shape is either inflated or constricted. Inflated means it is round. It is, filled with a lot of air and it looks, very strong. Constricted means it will not look healthy. Seed shape will be round or wrinkled. So it will be round like this or wrinkled. And seed color yellow and green. So these are the various characteristics, features of pea plant. That is the 7 contrasting traits. Now in your exam they will be asking you different characteristic features. So you should know this chart thoroughly. We should be prepared for any kind of question that can be asked in the board. So I would like you to concentrate on the letters that we are going to now assign. For example, if you are taking plant height and the DNA is double-stranded DNA, so then you have to give capital T and capital T. That is the uppercase of the first letter of the characteristic feature. So if it is dwarf, then you will be using small t, small t. Exactly the lowercase, just to differentiate. Now if you ask me, why don't we take capital T and dwarf D, that is D letter. It is not possible. Why? Because when you work out a cross that we are going to do later, it will, confuse the entire process. So we are not going to do that. Now flower color is purple and white. Here we are going to, this is the only exception, we are going to use capital W, small w, small w. And here we are going to use capital A, and small a, small a. And green is capital G, small g, small g. Inflated capital I, constricted small I, small i. Round is always capital R, small r, small r. And yellow will be capital Y, small y, small y. Students, don't forget, when you are writing this, you should make sure the upper and lower case letters are clearly distinguished in your cross in the paper. Now usually when we go to the next part of the lesson, we will be talking more about a gene, a chromosome, DNA and chromatin. Now you should know the differences between all these. So what is the meaning of chromatin, DNA, chromosome and a gene? You should understand this clearly. Otherwise it will be very difficult for you to answer questions that can be case based. So what do you mean by chromatin? Normally when you are looking at a cell through a microscope, this is how the cell will look like. You have the cell, then you have the nucleus, and then you have the DNA inside. So whatever that you are seeing inside this nucleus through an electron micrograph, this is how it will look like. This entire structure that you are seeing is called as chromatin. Now what is a chromatin? This chromatin is nothing but it is a mass of proteins that are present inside the nuclear cytoplasm, and around these basic proteins the DNA is wound. So for example, this is one basic protein, this is another one basic protein. These are all proteins, okay? So these proteins are there inside your nucleus, in the nucleoplasm. Now how the DNA will be wound around it? So the DNA is red in color, I am just showing you this. So this is how the chromatin is. Why this arrangement? Because we need lot of space to accommodate this long length of DNA. We don't have space. So for this you have basic proteins around which the DNA is wound. So this kind of structure is called as beads on a string structure. Beads on a string. So this is how the chromatin is present. Now you understand what is the meaning of chromatin. This is how normally when a cell is not doing any work, this is how chromatin will look like. Now what is there inside this chromatin? Inside this chromatin you have the DNA. The DNA is a double helical structure. This is the double helical structure of DNA. So this is how DNA you have to visualize. Now this DNA will change its structure when the cell is ready for division. When the cell is about to divide, this DNA will arrange itself in the form of chromosomes. So this is a chromosome. This is a pair of chromosome like this. There are many pairs. Depending upon the species, the chromosome pairs number will vary. For example, if you take humans, there will be 23 pairs of chromosomes. So this is how the chromosomes will be. Now what are chromosomes? These are the highly condensed structure of DNA. When the cell is about to divide, the chromosomes are present like this. So this is called as a chromosome. Now where exactly do you fit in the gene, the term gene here? Now you understand what is a DNA, what is a chromosome. Now where exactly is the gene? Gene is nothing but it is a segment here. For example, a segment of DNA here is called as a gene. For example, this DNA here is got from parent, that is mother. This is got from the father. This is responsible for a characteristic X. The same exactly at this position, it will have the opposite feature, or it can be the same feature. It is present on the chromosome or it is present on the DNA. A segment of DNA is called as a gene. Now I will tell you exactly what it is. Chromatin means it is the entire DNA that is wound around the basic proteins. And DNA is nothing but the double helical structure, and chromosome is the highly condensed form of a gene or DNA, and gene is nothing but a segment of DNA. So this is the basic thing you need to visualize before proceeding into this lesson. Now for grade 10, if you ask me whether you need to learn or understand all these terms, I would suggest that it is very, very important for you to understand these terms. You cannot directly jump into the lesson because these terms will be used here and there. And if at all there is a case-based question, I want you guys to, learn it clearly what exactly these terms means. So no harm in learning, right? So let us go into that. The first one is gene. So what do you mean by gene? Just now I told you it is a segment of a DNA. It was earlier called, during Mendel's time, it was called as factors. These were called factors. So what is a gene? This is the one which is carrying the information. Means it determines the biological character of an organism. We say that a person has got black hair. That means it is in his gene. That is a biological character of that particular organism. This gene is nothing but it is the functional unit of inheritance. This is the exact definition. Gene is a functional unit of inheritance. And it contains information required to express a trait. So any characteristic feature, if it has to express in an organism, this is how it will be present in the form of a gene. Number 2, we are going to talk about an allele. Allele means what? Different forms of same gene, and it codes for pair of contrasting traits. So like I told you, let us see this structure again. Students, listen. You have one gene that is inherited from the father, another one gene is inherited from the mother. Now if this gene is coding for X, I told you it can be like any character. For example, like I told you, black eyes. Okay, so this gene is responsible for black eyes. This is got from the mother. And here it may be for what? Brown eyes. This is got from the father. So it is present here in the same location, but father has got a different characteristic feature, a mother has got a different characteristic feature. But they both code for a single character. What is that? Eye color. But what are the two traits we are talking about? Brown eyes and black eyes. So this is what we are talking about here. Alleles, different forms of the same gene. The gene, the characteristic trait we are talking about is eye color. But the pair of contrasting traits that we are seeing here is black or brown eyes. That is what we call it as an allele. Next one is position of a gene on a chromosome. If you see here, this is the position I am talking about. So here the same position it is. Exactly opposite in the DNA. So this is the position of a gene on a chromosome is called as locus. Loci means plural, locus means singular. And trait, any characteristic feature of an organism is called as a trait. And allosome means sex chromosomes. That means if we take human beings, there are 23 pairs of chromosomes. In that 22 pairs are autosomes, otherwise called as body chromosomes. They are called as somatic. Plus they will have the last one either as XY or XX. If it is XY, we represent it as male. This is the symbol we use. If it is female, it will be represented like this. So these are the sex chromosomes, otherwise called as allosomes. There is a mistake here. Allosome and autosome. Autosome means body chromosome and allosome means sex chromosomes, which determines the sex of the organism.